What happens when your microbiome goes off the rails? Well, C-Difficile infection is a fairly common problem. What is C-Difficile, you ask? Well, it happens as a reaction to the consumption of some microbiotics. C-Diff for short is a species of gram-positive, spore-forming bacteria that accumulates in your gastrointestinal tract. The symptoms can be diarrhea, fever, nausea, and abdominal pain. Now, there are a number of treatments. Some work and some don't. And that's because the bacteria may have already developed a resistance to antibiotics. That can mean that a dramatic intervention might be required, and that dramatic treatment might be fecal microbiotic transplantation. Something you've heard about, but that doesn't mean it's readily available. And it's important to note that it's filled with a wide range of challenges. One of them, just as in organ transplantation, is that you need to have a match between the donor and the patient. Canada has been an early adopter of the procedure, and researchers are now asking, can it be applied in a number of other conditions, such as diabetes and obesity? These researchers are trying to determine if you can change the microbiome, can you then change the health outcome? We invited Dr. Jeremy Burton, the Deputy Director of the Canadian Centre for Human Microbiome and Probiotics, to join us for a conversation that matters about the exciting research underway in treating your well-being by re-engineering the bacteria in your intestinal tract.
Conversations That Matter is a partner program for the Centre for Dialogue at Simon Fraser University. The production of this program is made possible thanks to the support of the following and viewers like you.
Dr. Burton, welcome to Conversations That Matter.
It's a pleasure to be here.
You are specializing in microorganisms and their relationship with the body. It sounds fascinating, it sounds interesting, we hear about it a lot, but where are we really in understanding the interplay between the microbiome and our overall health?
I think we've realized now that the human microbiome is incredibly important to sort of every health system in the body, essentially.
Not just the gut.
Not just the gut, far beyond the gut, every health system just about, from the brain to the urinary system. So we're now interested in, how can we change what's going on in the microbiome for benefit for people? And so we don't really have a lot of tools in order to do that, and very crude tools. So diet's quite important, but sometimes the microbiome has gone beyond a point of no return with regards to getting a return, from a diet-based intervention. So we need to do something quite extreme. So that's where fecal transplants have come in. So people don't really exactly understand what fecal transplants do. Obviously, it's the transfer of fecal material from one person to another, and that transfers certain traits, which we've shown that scientifically. But actually, how does it work? We don't exactly know. Is it that we're transferring a total ecosystem from one functioning human to another human that is less functional? All the different bacteria. That's a potential case, but other people have done studies which suggest that you don't need to have all the bacteria alive, or that you only need to transfer a portion of that microbiome to get a clinical success. So we don't really fully understand what we're doing, because it's an incredibly complicated system.
Well, and I guess that's why it's not readily available either. It's an idea. There are tests that are being done, but it's not something that you can say, I want to go have this done.
No, that's right. So it's obviously had its success in people with recurrent C. difficile infection, where
explain what C. difficile infection is, just for anybody who might be going, what?
So quite often when antibiotics are used, sometimes you get a bacterial overgrowth of some bacteria that aren't affected by the antibiotic, and the microorganism Clostridium difficile, which we sort of just call C. diff, is one of those bugs that can overgrow. And sometimes that can't be treated with antibiotics, and it just keeps...
Because it's a byproduct of antibiotics to begin with.
That's right. It has resistance, and it's quite it doesn't respond well to antibiotics, basically. So I've lost my train of thought.
Well, and so you're saying we wind up in a situation where somebody might be experiencing C. difficile, and that the fecal transplant may actually be able to help them because antibiotics won't necessarily address now what has become the byproduct of an antibiotic reaction.
That's right. So we need to do something dramatic, and so that dramatic is transferring, the whole fecal contents of one person to another. And there's some use of this throughout history because it's, a relatively crude, thing to do. So I think German troops used it in the First World War, and other Bedouin tribes have employed this technique. So it's a concept that isn't brand new, even though it's sprung into, the height of, medical interest at the moment. So
it's become fashionable.
Fashionable, yes. I mean, it's used. It's had a lot of use in this C. diff field because those patients which have recurrent C. diff infection are suffering from diarrhoea and are very sick, and eventually that may lead to a scenario where they have to have their colon removed or even death. So a pretty good alternative to those options is to actually have a fecal transplant.
I just got to get you to hang on for a second while we take a quick commercial break. We'll be right back. Sorry for the interruption, but I need your help. I'm asking you to support the show by becoming a patron. That means pledging $1 per show. Conversations is a unique program. There's no grandstanding, no arguing, no yelling. Instead, I sit down with people who have unique insights, specialized knowledge, and experiences that inform and enlighten. And we do this on an incredibly limited budget with a very small crew. Well, this is my office, and it's where I edit the show. And then once it's ready, we give it away. So many different websites around the world are carrying this show, but they get it for free. They don't pay us. So I'm asking you to pledge $1 per show by going to patreon.com/conversations that matter. Thanks. We want to keep producing conversations, and with your help, we can. Now let's get back to the show. So are you telling me that the fecal matter of one person will, just by its very introduction, will become the dominant, environment just by the introduction of it? Because it's not like it's going into a totally void environment.
That's correct. So you are putting this material into, where another microbiome exists, but you're taking a whole consortium of bacteria with you. And it doesn't always become dominant. It depends on the person. Quite often it's a merge between the donor's microbiome and the recipient's microbiome. And often the changes are, over time that it reaches a sort of a happy balance between the two microbiomes, and in the process hopefully restores whatever the clinical problem is. So it's been very successful for this illness, and Canada has been an early adopter of this procedure. So because there's so many other diseases now that we associate with the microbiome, people were wondering, can we use this therapy for some of those conditions? And there's links now between the microbiome and obesity, diabetes, a number of conditions. And so if we can change the microbiome, maybe we can change those people back to a more healthy state.
But do we know that we can do that? At this point, do we know whether or not those, organic and biological mechanisms are going to work?
No, not exactly. I mean, the people have done a lot of animal studies, which is why they're interested. So they can take the microbiome out of a obese mouse and put it into a lean mouse and not change anything else and end up with a obese mouse. So we know from animal studies that you can transfer phenotypes. So the use for other applications in humans has been fairly limited to date. There's been some Dutch and other studies which show that you can help in some of these metabolic syndrome cases, and that's really sparked the imagination of a lot of other studies, basically, because there's so many that have some sort of gut link, from MS through to diabetes. So people were interested in harnessing it because the other methodologies such as diet and probiotics, antibiotics, and other treatments haven't been successful.
So it's my understanding that right now the greatest amount of work that we've done is in the gastrointestinal tract. I would imagine that if you are suffering from irritable bowel syndrome, you would be saying, okay, is this going to work? Please.
There's studies emerging now. Some have had mixed results that suggest that it can work in some of these conditions. But each person's problem is unique in its own way. We all have a unique microbiome. So I wouldn't like to say it's the be-all and end-all for those patients. I think that there could be a mixture of treatment that's required, and so it's very early days and we need to do some more thorough studies.
Well, I would imagine it's incredibly complex. I think back to the 1990s when the race was on to complete the mapping of the human genome. And I noticed that from your own research that Craig Ventnor is one of your sponsors, and he was instrumental in the mapping of the human genome. Is this as complex as trying to unravel our DNA?
So, so the Craig Venter Institute is an institute in itself, and Craig Venter obviously headed that institute. So we work with people in that institute, and they've provided assistance for us. This is a far more complicated problem than just looking at the human genome itself.
More complicated.
More complicated.
Because we're talking about 13 billion bits of information in the human genome.
Well, that's right, but we've got our combined functional genes from the microbiome is again 15 times what the human amount is. So not only do we have to work out what the interaction between the human genes are, but all these new microbial genes that we've ignored previously.
Oh my gosh, so we have to understand their RNA as well.
Their DNA, absolutely.
DNA,
absolutely. And try to work all that out. Each different microorganism is almost a bioreactor in its own right. And so, there's an incredible complexity. And each person has a, very unique microbiome. So infinite combinations of genes and metabolites and factors that have to be, taken into consideration. So, we can already get sequences of the whole microbiome. The advantage compared to the original Human Genome Project is that sequencing technology has advanced quite dramatically. So
we know how to do it, yes.
So we can do it a lot cheaper now because the original, I can't remember what the Human Genome Project cost. It was something like $5 billion.
Yeah, I think it was a little more than that.
It was expensive.
Yeah.
Now you can sequence the same human genome for maybe a few $1,000s, maybe even cheaper. And so now we can apply some of that technology to sequencing the samples for microbiome, and we apply that technology. So we're getting a lot of data, and part of the problem is handling that data and comparing that data to all the other things going on. So that's where a lot of the technology for this comes in.
I did a talk a couple of years ago where Jeffrey Bland, the father of functional medicine, was talking about the fact that we're not very far away from, I'll show up at the doctor's office saying, here's my sequenced DNA, so that you've got a pretty good idea of what my genetic structure is. Are we now also looking at, and here is my microbiome? Are those days coming? And what would be the value of knowing what the microbiome is in advance?
Yeah, I think those days are coming. And if you think of the human genome data, well, it's the markers, single genes that were found of real interest, which they might look for in certain genetic tests. And there probably will come a day when you have your microbiome assessed, or even certain markers of the microbiome. So there's been some really good research studies which show the human microbiome metagenome, so all the genes in the microbiome, a very good indicator of human health for certain conditions, metabolic kind of conditions like type 1, type 2 diabetes.
Yeah.
So it's sort of like the canary down the mine. It's a good indicator of the health of the overall system. And I don't know if we should be, we treat the microbiome as separate to ourselves, probably because of our daily activity to get rid of some of it. But in reality, we should all be treating it as one thing, because it's almost an organ in its own right. It provides us, a whole lot of useful metabolites and vitamins and material. So we need to start treating it as an organ, and it gets a lot of abuse, and we need to look after it a little bit better.
But in essence, we're talking about bacteria.
That's
and
right.
that is like coursing its way through our body. I had heard that there's more bacteria in us than there is us.
Yeah, there's more than probably 10 times the amount of bacteria in us. But obviously it occupies a smaller amount of space. But each one of those microorganisms is busy doing something or producing something, and it's a close relationship with the host. There's an interaction. We need microbes to develop our immune system, to get certain nutrients, to break down food. There's a zillion different things that they do for us. We have a close relationship, and we even think that we regulate the microbiome to perhaps produce those things on demand. It's the microbiome's been shown to be important from daily circadian rhythm to, coronary heart disease. So it plays a role, and we have a close relationship, and we need to understand that relationship a lot better.
Right. So is it true that if I have a particular kind of diet, I am now starting to attract those microbes that I will need to be able to process that food?
Well, you're going to attract the microbes that like to eat what you've just eaten, and that's they're a little bit selfish in that way, and so hopefully that you've got the microbes that are also good for you. So, one of the biggest risk factors for cancer is diet, and we know associations between things like red meat and the microbiome and cancer. So, we select the microbes by what we eat, and we can change our diet and hopefully improve our microbiome, because when you're eating a lot of meat, you're selecting microbes that break down the substances in meat, and those substances, when broken down, are carcinogenic.
Right. And they carry with them, in most cases, some sort of antibiotic, which sort of leads me to my next question. We get ill, we have a gastrointestinal infection or something like that. We treat it with antibiotics.
Yeah.
And now we're out of balance. What role do probiotics play in helping us to reestablish that balance?
Yeah. I mean, it's a bit of a fallacy that we reestablish a balance or we recolonize with probiotics.
But that is the popular opinion.
That's true, but that doesn't really happen, because probiotics typically go in and then they come out again. But they do seem to work. The evidence for improving antibiotic-associated diarrhea isn't too bad. And we have to remember that that the probiotics don't colonize, so they're working by another means. So, you could be taking that probiotic while taking the antibiotic as well, because there's other things with the probiotic that come with it, like during the fermentation process, it produces metabolites and other things. The immune system also reacts to cell components, so the bacteria don't necessarily have to be alive. You might just need a small part of what is with that probiotic to get some benefit. So there's a number of mechanisms at play, but I'll tell you what doesn't happen, that is, you don't replace your bacteria with the probiotic bacteria typically.
There is this whole, take this probiotic, have this yogurt with a probiotic in it, or through a variety of other methods. Is it still worthwhile if it's really only doing a small part?
Oh, absolutely. I think it's well worthwhile, and studies show that. It's just that we don't fully understand what it's doing. Just because it doesn't work the way we think, as long as it works is the important thing.
How much do you take? What's the best source? Do you take probiotics? And if so, what, where do you get that from? Like what?
So, how much should you take and which ones should you take depends on what your condition is. I'll tell you one thing is that you can't take too many, and
you can't take too many.
You can't take too many. They're pretty harmless, and
you're not going to OD on probiotics.
You're not going to OD on probiotics, and as someone in this type of research, I've eaten a lot in my time.
Yeah.
The best place to find information about probiotics is there's a Canadian probiotic guide, which is available on the internet. It looks at what's available on the market, what the evidence is, what the dose is, and what you should take.
Huh, okay. So you would recommend that. People who are watching this, of course, are going to be fascinated by this. There's going to be some people who are going to say, I don't want to know anything about it. But what role do fermented foods play? Do they also help in fostering an appropriate environment for the microbiome?
Yeah, I think that it does. The evidence is a little bit sketchier with fermented foods because a lot of them don't have proven benefits. They've never been tested. Not all of them have been tested in clinical trial situation. And
it's interesting days as far as our understanding about the microbiome. I gather that we're going to continue to have one revelation after another over the next decade or two.
Yeah, I think this is the hottest topic in science at the moment, and people sort of viewed it as a fad, but it doesn't seem to be slowing down. For someone who's worked sort of in the field for most of my life, I find it kind of quite surprising because the field has shifted quite dramatically.
Yeah.
But if we're finding such a close relationship with humans and our health, we're going to have to really understand the microbiome. And so there's plenty of fertile ground for future study.
And there appears to be a brain-gut connection as well.
We see this in quite a few studies now. So bacteria binding GABA receptors, which are linked to the vagus nerve, which interact with the brain. People have taken feces out of people with irritable bowel disease, and some of those patients have anxiety as a comorbidity in that problem, and they'll transfer it to a mouse model, and they'll actually see the anxiety transferred to a mouse. So this is an incredibly powerful thing that we're dealing with, and although we can potentially transfer benefits to people, we also have to be extremely careful that we're not transferring something adverse, something that we don't even know that is there, something that might be in our future, something that, like, you might have a cancer or a heart disease in your future and you don't know, but maybe the microbiome already knows. It's very sensitive to some diseases.
Wow. Well, thank you for coming in and shedding some light on this.
Thanks for having me.
My pleasure.